Welcome to This Explainer. Today, we're diving into what is honestly the ultimate detective story in all of molecular biology. We're going to track the journey of a single microscopic strand of RNA. We'll follow it literally from inside a living cell all the way to a physical lab bench, into the digital brain of a supercomputer, and finally right into a hospital clinic where it's actually saving lives. So let's just jump right into this fascinating journey of the cellular messenger.
To unravel this mystery, we've got a really clear roadmap today. We'll start off by decoding these messages of life, zooming in on the forest versus the trees, preparing our RNA library in the lab, running the digital bioinformatics pipeline, finding meaning in the data, and finally, looking at some real-world medical breakthroughs.
All right, Section 1, Decoding Messages of Life. Before we can sequence anything at all, we really need to understand the foundational why behind this whole scientific field.
So think about this for a second. How do we actually read the active instructions of a human body? Well, think of your DNA like the permanent blueprint of a house, right? It's locked safely away in the nucleus, which is basically the manager's office of the cell. But, a blueprint doesn't actually build anything on its own. Instead, the cell makes these temporary copies of specific instructions. It's kind of like handing out little post-it notes to construction workers for their daily tasks. Those temporary post-it notes, that's RNA. And by reading these RNA messages, we don't just see what a cell could do, we see what a cell is actively doing, right in this exact moment.
Now, all of those temporary post-it notes combined make up our very first major concept today, which is the transcriptome. The transcriptome is the complete, dynamic set of RNA transcripts floating around inside a cell at any given time. And the cool thing is, while your DNA genome is pretty much static, it really doesn't change, your transcriptome is constantly shifting. It changes based on stress, development, or even disease. Decoding this exact snapshot is literally how scientists are diagnosing rare genetic diseases and figuring out how to fight specific cancers. It's an absolute game changer.
Moving on to Section 2, the forest versus the trees. Because when it comes down to looking at these transcriptomes, it's really all about scale.
To put this into perspective, think of bulk RNA sequencing like looking at a massive forest from an airplane window. You get the overall average color of all the leaves, which is great for a big picture baseline, but it completely masks the rare individual trees down below. Single-cell sequencing, on the other hand, well, it lets us walk right through that forest and examine every single tree individually. It resolves what we call cellular heterogeneity. Basically, it allows us to spot that one rare, maybe mutated cell type that's just hiding out in a massive crowd of totally healthy cells.
But wait, what happens when you have a tissue sample that is just way too tough to gently separate into those individual cells? I mean, picture a frozen post-mortem brain or a piece of tough, beating heart muscle. If you try to just pull those apart, you'll literally destroy the delicate cells. And that is exactly where single-nucleus RNA sequencing, or SNN-RNA Seq, comes to the rescue. Instead of trying to isolate the whole, fragile cell, scientists basically just extract the hardy nucleus to read the RNA locked inside of it. That way, we preserve the biological integrity of the whole sample. Pretty clever, right?
Okay, Section 3, preparing the RNA library. We're now taking our journey out of the theory and straight to the physical lab bench.
I want you to imagine this preparation as a highly sophisticated factory shipping process. First up is cell lysis, which just means we break the cell open. Second, we capture the mRNA using these things called poly-T primers. Think of this like using a microscopic molecular magnet that only grabs the very specific RNA posted notes that we actually want. Third, we use a process called reverse transcription to turn that super fragile RNA into stable double-stranded cDNA. And finally, we add UMIs and barcodes. UMIs are basically like unique serial numbers for every single molecule, while barcodes act exactly like zip codes, tracking exactly which specific cell that piece of RNA originally shipped from.
Now, during this factory process, we often use a really clever trick called the DUTP strandedness method. You might be wondering, why does strand direction even matter? Well, remember, DNA is double-stranded, and sometimes genes actually overlap each other on opposite strands. If we just mix everything up into DNA, we completely lose the information of which side the original RNA came from. So by using DUTP, we leave a little molecular breadcrumb trail, if you will, that preserves this directional information. It makes absolutely sure we don't get confused when we're trying to read those overlapping genes later on.
So, once our library is all prepped, we have to actually read the letters. Usually, we're choosing between short read and long read sequencing. Short read is kind of like taking a pair of molecular scissors and chopping the RNA into tiny little fragments. It's incredibly affordable, and it's perfect if you just want to count how many copies of a certain gene are present.
But if you want to see the full, complex structure of a transcript, like, say, finding novel splicing isoforms, where the cell actually rearranged the instructions, well, then you need long-read sequencing. That method reads the entire molecule from end to end without chopping it up at all.
Section 4. The digital bioinformatics pipeline. Our cellular messenger has been chemically processed and read by a machine, so it's time to transition from physical lab tubes straight into the digital realm of supercomputers.
Now, every RNA-seq analysis out there relies on a universal 5-stage pipeline. But here's the catch. If we hit a stop sign at step 1, which is quality control, literally nothing else matters. Before trusting any data, scientists have to look at what are called Q-scores to ensure the digital letters aren't corrupted. They use software tools to check for adapter contamination, which is basically just leftover molecular packaging from the library prep, and they trim it all away. If your median Q-score is too low, or if you have way too much ribosomal RNA contamination, your digital data is basically garbage. You literally have to stop right there.
But let's say the data looks good. The crucial point is how we handle step 2 of the pipeline, alignment. You have billions of these short digital text fragments, and you need to match them up to a reference human genome. Think of it as 3 incredibly clever ways to solve a massive digital jigsaw puzzle. You could use a tool called STAR, which is like building the puzzle while carefully looking at the picture on the box. It's highly accurate, but it takes hours and a ton of computer memory. Or you could use string tie, which builds the puzzle completely blind from scratch, specifically to discover brand new novel transcripts. Or, option 3, you can use Salmon. Salmon is a lightweight tool that basically speed reads just the titles on the puzzle pieces. It takes minutes and very little RAM, making it perfect when you already know exactly what you're looking for.
All right, Section 5, Finding Meaning in Data. Because having billions of mapped letters is great and all, but biologists actually need to turn this massive digital puzzle into usable biological meaning. To make any sort of fair comparison, we have to level the playing field. Think of it like balancing a scale. This mathematical correction is known as normalization. Why do we need it? Well, because you might have accidentally sequenced one sample twice as deep as another. We use something called TPM, or transcripts per million, to fairly compare the expression of different genes within a single sample. But if we want to compare expression between different samples, like say comparing a healthy cell to a sick cell, the gold standard is to use DSeq2's median of ratios method to mathematically balance that scale.
Once everything is nicely normalized, we use some incredible statistical tools to basically hunt down genes that change significantly between healthy and diseased conditions. This is what we call differential expression analysis. DSeq2, the one I just mentioned, is absolutely fantastic for small sample sizes and for controlling false positives. Then you have EdgeR, which is a bit more sensitive if you have larger groups of samples. And finally, LimaVoom is highly efficient for incredibly complex experimental designs. But remember this golden rule from the source material. You always need a minimum of 3 biological replicates per condition if you want to actually trust your statistical results. 1 or 2 just doesn't cut it.
But hey, even with the absolute best math in the world, there is a massive warning we have to heed here. Beware of batch effects. A really common, honestly devastating mistake is processing all your healthy samples on a Monday and then all your disease samples on a Friday. Just the slight temperature difference in the room on those 2 days or using a new batch of chemicals will create artificial variations that completely mask the real biological discoveries. It's wild. You must always randomize your sample processing for sure.
Section 6, Real-World Medical Breakthroughs. We've gone from biology to chemistry to computer science, so let's bring this explainer home and connect this dense, multi-step journey directly to human health.
When we put all of this together, the clinical applications are honestly truly amazing. Standard DNA sequencing sometimes hits a complete dead end when doctors are trying to diagnose rare genetic disorders. But by using RNA Seq pipelines like one called DROP, doctors can actually find weird, rare RNA splicing events that standard DNA sequencing missed entirely. This literally saves patients from agonizing, years-long diagnostic odysseys. In oncology, reading the transcriptome helps identify dangerous gene fusions so doctors can tailor highly personalized cancer therapies. And in pharma, scientists are now screening hundreds of drug treatments in parallel, reading the RNA to see exactly how a cell responds to a potential new medicine before clinical trials even begin.
There's a quote from the source material that really hits the nail on the head here. It says, "Clinical RNA sequencing serves as a powerful complementary tool identifying functional defects such as aberrant splicing, gene fusions, allele-specific expression, and extreme expression outliers. Basically, what this is telling us is that RNA sequencing is no longer just some neat research tool for academics sitting in a lab. It is fully evolved into a frontline diagnostic lifesaver right there in the clinic.
Which leaves us with this final, rather provocative thought. We are right now entering an era of spatial multiomics. This is where we don't just read the RNA, but we map exactly where it is physically located in a 3D tissue sample. As this technology continues to mature, I want you to imagine a future where a routine doctor's visit involves dynamically reading the real-time instructions of your very own cellular messengers. Just think, how will that level of personalized medicine change the future of your healthcare? Well, thank you so much for joining me on this journey through the transcriptome. Keep asking questions and keep exploring.